My name is Elena Macías Sánchez and I am Ramón y Cajal Group Leader at the Institute of Parasitology and Biomedicine López Neira. Our group is called Molecular and Structural Basis of Bone Mineralization, and it is dedicated to increase our knowledge on the mechanisms that drive bone formation and regeneration and how these mechanisms are altered during pathological conditions. Our research is defined in three main pillars. We aim to understand the role of mineralization regulators, the so-called non-collagenous proteins and proteoglicans that trigger the mineralization in vertebrate skeleton. We also evaluate the structural and molecular changes associated with the abnormal expression of these regulators, specifically at the mineral-collagen interface. And we apply biomimetic approaches directed to the development of osteocompatible materials for biomedical applications. Our work is focused on discerning the role of certain molecules in the initiation of mineralization. Specifically, we are currently working with a type of proteins which are called proteoglicans, since they have a protein core and several covalently attached sugar chains that seems to have a role in facilitating the deposition of the mineral. We aim to discern the role of these regulators and understand the effects of their absence during pathological conditions to be able to design therapeutic approaches for these pathologies. The type of questions we face nowadays are complex in nature and transcend a single field of study, and therefore need for a collaborative approach that involves the participation of different disciplines. In the case of bone, we deal with a complex hierarchical material, a tissue, and an organ. And if we want to really understand the formation and degradation processes, we need to integrate all these aspects as a whole. We use a multimodal approach combining the use of advanced electron microscopy techniques, spectroscopy, and X-ray tomography and diffraction, with traditional biochemistry and cell biology methods. In this sense, our goal is to bridge the gap between the material science perspective and the biomedical research, and to find ways to transfer this knowledge to the clinic. We have contributed to the identification of a new hierarchical level of organization in bone called tessellation. There has been a long-lasting debate about the progression of mineralization in collagen-based materials related to the location of the mineral crystals, inside or outside the collagen fibrils, and how the mineralization front advances in the structure. Only recently it has been proposed that the mineral may propagate in a continuous manner through the extracellular matrix, forming a mineral network. We now know that the mineralization takes place in a continuous way, but forming discrete structures called spherulites or mineral ellipsoids. These structures are small mineral ellipsoids that grow until they merge, forming a three-dimensional tessellated structure. And this structure has an important role in the mechanical properties of bones, since it is able to reduce dramatically the propagation of cracks in the structure. Well, with an aging population, degenerative skeletal disease represents one of the major social and health challenges. I think the most prominent example is osteoporosis. There are 22 million women and 6 million men diagnosed in the European Union alone. Skeletal disease causes the greatest amount of premature death and disability in the European Union and worldwide. However, despite their social and economic impact, the mechanism of bone formation and regeneration remain largely unknown. So there is an urgent need to understand the mechanisms that determine bone mineralization and degradation, and to translate this knowledge to the clinic.